Hello everyone! In this video, I will show you how to use our 3D yeast tool when presenting to customers, whether it is during a Yeast Academy, at a trade show, or in a face-to-face meeting. This 3D tool has been designed to make the yeast story simple and visual. Each piece represents a specific fraction of the yeast cell, and by taking it apart step by step, you can demonstrate how every part of yeast has its own role and value. The objective is to help you structure the story and guide the customer through the tool using the pieces in the right order. This is the yeast cell. It contains two main parts. The intracellular content, which is the soluble fraction, and the cell wall, which is the insoluble fraction. First, let's look at the wool inactivated yeast, also called dried yeast. It contains all the constituents of the yeast cells. Inside, proteins, mostly in the form of large peptides, vitamins, and minerals. Outside, the cell wall, which is rich in polysaccharides. But when the yeast is simply dried, the cell wall stays intact. That means most nutrients remain locked inside and cannot be efficiently digested and absorbed by the animal. As a result, wool inactivated yeast works mostly as a basic nutrient source and palatability enhancer, but with a low digestibility and limited functionality compared to more processed yeast, such as hydrolyzed yeast. Here, with the hydrolyzed yeast, specifically selected enzymes are used in a controlled process to gently open the yeast and make the intracellular content available. Hydrolyzed yeast still contain both the cell wall and the intracellular content. There is no separation step. The result of the hydrolysis process is that protein and nucleic acids are converted into small peptides and free amino acids and also nucleotides. And hydrolyzed yeast still contains vitamins and minerals. Nutrients are highly digestible, amino acids have fast absorption kinetic, the composition is consistent and reliable, and it also acts as a palatability enhancer. So hydrolyzed yeast delivers the full benefit of yeast, both nutritional and functional, in the most efficient way. Now, if we take hydrolyzed yeast one step further, we can separate the two fractions. The insoluble fraction, which is the yeast cell wall, and the soluble fraction, which is the yeast extract. Yeast cell walls represent 30 to 60% of the yeast dry weight, depending on the type of process and the quality of the separation. There are two main polysaccharides: mannose oligosaccharides in the outer layer and beta-glucan in the form of beta-1,3 and beta-1,6-glucan, which is the inner layer. These functional ingredients have clear benefits: to bind undesirable bacteria, thanks to mannose oligosaccharides, such as E. coli, salmonella, preventing adhesion in the gut, to help reduce the effect of some mycotoxins, for example, fumonisin and zearalenone, and to interact with the immune receptors like dectin-1 with beta-glucan and TLR with the mannose, stimulating immune system and reinforcing natural defenses. This is why yeast cell walls are widely used as functional ingredient to support gut health, immunity, and animal performances. Beyond yeast cell walls, it is possible to isolate beta-glucan. These are concentrated fractions of beta-1,3 and beta-1,6 glucans, obtained through specific separation processes. Purified beta-glucans are often used in situations where a strong and reliable immune system is needed, for example, under stress or during disease challenges. This makes purified beta-glucans a very specialized functional tool. Yeast is a teeny cell, but it carries a powerful story to tell. This 3D tool helps you share that story in a clear and visual way, showing step by step that each part of yeast offers a specific advantage and that every part should be used in the right situation to support animal health and performance.